This is the G protein. It acts as a molecular switch in this process. Whether it is on or off depends on which of the two guanine nucleotides are attached, either GTP or GDP. This protein relays signals from the plasma membrane signal receptors to other signal transduction proteins in the cell. This is the signal molecule. It binds to the extracellular side of the receptor, causing it to activate and change shape. This is a plasma membrane receptor that works with the G protein, otherwise known as the G protein-linked receptor. Membrane receptors transmit information from the extracellular environment to inside the cell. This is an enzyme. The enzyme works with the G protein and the receptor. They are important in cellular response as they trigger the next steps in the pathway once they become active.
Initially, GDP is bound to the G protein, which means the G protein is inactive. The G protein is loosely attached to the cytoplasmic side of the membrane. The G protein and the receptor work together with a protein, which in this case is the enzyme.
When the corresponding signal molecule binds to the G protein-linked receptor, it becomes activated and changes shape. This occurs on the extracellular side of the receptor. On the cytoplasmic side, the receptor binds with the inactive G protein. This causes a GTP to displace a GDP, and the G protein becomes active.
Then the signal molecule leaves the G protein-linked receptor and the receptor becomes inactive. The active G protein dissociates from the receptor and diffuses along the membrane. Then it binds to the enzyme, causing the enzyme to become active. This leads to a cellular response as the active enzyme can trigger the next step in the pathway.
The G protein acts as a GTPase enzyme and hydrolyzes GTP to GDP. Then the G protein becomes inactive and leaves the enzyme. Then the G protein can be reused to start the process over again.